So the next method is MNN correct. This method is actually developed to merge multiple data sets that examine the same biological system. So it assumes each batch you're considering shares at least one biological condition with every other batch you're considering. So this isn't, in a balanced design, that would be the case, because each batch would have some from every biological condition you're considering. In our case, because we have replicates, that's not the case. So in order to use this method, we have to consider each individual separately and consider all of the batches for that individual and correct them using this method, and then go on to the next individual and correct their batches separately. So in order to do that efficiently, I've defined this function where we select each of the batches separately, and then we apply MNN correct to each of those batches, and then it uses some parameters. So MNN correct is based on mutual nearest neighbors. So it takes, for every cell, it finds the mutual nearest neighbors in the other batches to that cell, and then calculates the distance between those, and then subtracts that in order to correct the batches. So to calculate the mutual nearest neighbors, you need some sort of number of nearest neighbors you're going to consider. So that's k in this case. So we set it at 20, and then we have a couple of other parameters that I'm not going to worry about for now. This is just a normalization method that we tell it to use. And it's dimensionality reduction parameter for pre-processing the data. And then we have to do this complicated if statement because we have one individual with only two batches, because we removed one batch during normalization. So that's all this is doing, is for that individual where there's only two batches, only consider two batches. And then we apply that function to the subset of the data for each individual. So that's individual 1, individual 2, individual 3. And we combine all three back together again at the end. Whereas if you had a balanced design, you could simply separate each of the batches and apply this method to each batch. You don't have to tell it the biological conditions. It will figure that out for itself. And then it would be a single command. And the last method we're going to consider is just a general linear model, which is essentially a simpler version of Combat. However, it's easier to customize than Combat is. So here we just, again, because we have the confounded design, because we have replicates rather than balanced batches, we have to apply the GLM to each individual separately in order to preserve the biological variance and not remove it along with the batch effects. So again, I define a little function. Here we model the expression of our gene G as the product of the batch and the individual, and calculate we have to set the intercept to 0 just to make it easier to subtract the effects afterwards. And then here we're just applying this function to the normalized matrix, and here we're telling it the batch is batch, individual is individual. And it's applying it to each row separately. And then here we're actually, this is our matrix of estimated effects for each batch. And here we're using the factor of the batch to select the appropriate one for each column. And that creates our correction matrix, which we subtract from the normalized matrix. So here we're applying it to the entire matrix. So in this function, you can see we applied it to the entire matrix. So this will remove both the effects of the individual and the batch effects because of the replicates. And we have as an exercise to perform this correction on each individual separately, as we did for MNN up here. However, I think I'm going to skip that exercise and leave that as homework for you. We're going to move on to how to compare whether our batch effect removal methods have worked properly. The simplest and easiest way is to just look at the PCA plot as we did for the normalization. So here's our little function to plot our PCAs for all the different methods. So here's the raw counts. You can see, again, the library size is the dominant factor. And then when we normalize, for library size, we have the different batches showing up. And then if we use RUVG, we can see that we have gotten rid of some of the batch effect, but we've also gotten rid of some of the biological effect, because these two individuals are now overlapping a lot, which agrees with what we know, what some people have seen before, that spike-ins aren't truly independent of your biological effect for sort of unknown reasons why this is the case. And if we remove even more factors, this gets even worse. The batches look better, they're more mixed up, but so are the individuals. So we're losing a lot of the biological signal as well as the batches. Whereas if we use RUVS, using only one factor doesn't do very much. However, when we increase the number to 10, this looks pretty good now. Each individual is a separate group, and all of the batches are intermingled with each other. Whereas when we use Kaybet, you can see that it's gotten rid of both the batch and the biological effect. And we've now just got a big clump of cells with no interesting signal left. This is the same as if you account for the library size, it looks mostly the same. Doesn't make a big difference. If we use MNN, because we normalized each individual separately, we get very, very nice tight clusters for each individual that are extremely different from each other. And the batches are mostly overlapping. But you can see the means, and particularly in this individual, the means of each batch is slightly different from the others. So it hasn't completely gotten rid of the batch effect, but it's a lot less. Here's the global GLM. You can see it looks exactly the same as Kaybet. Whereas when we apply the GLM to each individual, we get something quite similar to MNN and RUVS. And there's another exercise, which I'm not going to cover right now. Yep.
Each of these techniques kind of has its time and place to use. Is there one that is just much better than the others?
None are particularly better than any of the others. Sort of depending on what your experimental design is, you might want to use one versus the other. In general, if you have unbalanced batches, so if you have one batch that contains a couple of the biological conditions, another batch that contains some of the biological conditions, and some of them overlap but not all of them, then MNN correct works quite well, because it can figure out which parts match between those batches and use that to estimate the correction. Whereas if you have a balanced design, pretty much all of the methods work really well. And if you have replicates, really only RUVS works well just straight out of the box. You can see the others, we had to sort of break down our matrix and apply them in a sort of weird way, whereas RUVS can handle replicates. So we also look at the cell RLE. However, this mostly looks at library size rather than specifically batch effects. So you can see once we get to the library size normalized data, it doesn't really make any difference when we applied the different batch corrections, because this method can't detect that. However, we can use the analysis we looked at before to break down the variance explained by each of the different factors again and look to see whether the variance explained by the batch effects has indeed decreased. So there's our raw matrix log, and you can see once we've done the normalization, for library size, the total counts have shifted way to the left and left just the batch effects and individual. If we then look at after M&N correction, we can see that batch effects has decreased a bit, whereas the individual has been left alone. I think these have the wrong titles. Oh yeah, this is our REVG. So this one has not corrected for the library size that well and has left the batch effect quite a bit, but the individual is also still there. REVG 10 looks pretty much the same. REVS, you can see here that the batch effect has been decreased a little bit, but not very much. Whereas when we look at REVS with 10, now the batch effect has been shifted quite a bit to the left. If we look at Kaybet, now both the individual and the batch are way over here, and they've been really completely removed, so that's not very helpful. Same with Kaybet accounting for size factors. See, M&N correct, here the batch effect is almost even with the individual, which is actually the best case scenario we can get. Because batch is confounded with individual, you should never see the batch become less than the individual in terms of explanatory power. So equal is as good as we can hope for. So this is almost perfect for M&N. For the global GLM, you can see again everything's gone. There's almost no signal from anything left. Or if we do GLM individual, here you can't even see the curve for batch because it's completely overlapping the curve for individual. Hopefully on your screens you can see that this orange curve is sort of brown because it's a mix of the blue and orange. Oh, and the last method is Kaybet. So another way of thinking about batch effects and how effective we've been at removing them is if you take a local group of cells that are all close to each other in high dimensional space, then the batch associated with those cells should be the proportion of cells from each batch within that group should be the same as the proportion of batches in the entire data set if there are no batch effects. They should be completely well mixed and completely random. So Kaybet is a method that essentially tests this by taking KNN networks, so the K nearest neighbors around each cell, and testing whether the proportions of each batch match the expectation given the frequency of batches in the entire data set. And then uses a binomial distribution as the test, and depending on how frequently that test is rejected in different parts of your data, will indicate how strong a batch effect is still there or not. However, it should be noted that that doesn't consider at all whether your biological signal is still there or not. It only asks whether you've got batch effects. So, however, we have to apply this per individual because we don't expect the batches from one individual to overlap, to mix with the batches from a different individual. So we have to apply this to each individual separately again. So this is how you do that. Yeah, so we've got the batch, and then we've got some parameters. You can look at that paper if you want to know the details. And almost on time. And then this is how you plot a nice, pretty picture of this. And here's the picture we get at the end. So here are all our different normalization strategies and whether they pass Kaybet or not. So you can see red in this case is good. There's low batch effects, whereas blue is bad. There's high batch effects. See, most of the methods score pretty well on this case, except for sort of RUVG don't score very well. And the raw transform data score quite poorly, whereas most of the others are improving things, except for this individual. However, it's interesting to note that you actually get really low score for batch effects in your raw counts. This is because the variance in the raw counts is dominated by the total counts rather than by the batches. So that's obscuring the batch effects in the total counts. So just because you get a low score here, a good score by Kaybet doesn't mean there aren't batch effects. It just means something else is more important than the batch effects. And then, again, his homework is to run the same analysis for the reads.